As an example where spectroscopy in the visible spectral range made important contributions to the understanding of a protein, we will now discuss Bacteriorhodopsin. Bacteriorhodopsin is a small integral membrane protein which functions as a light-driven proton pump. It converts light energy into a transmembrane proton gradient. The proton gradient is converted by the ATP synthase into ATP. Bacteriorhodopsin is found in the cell membrane of salt-loving microorganisms, archaea. It provides them with an emergency generator of energy when the oxygen concentration is low and respiration ceases. Bacteriorhodopsin is one of the proteins which are most intensely studied, for several reasons. First, it is a model for an important class of membrane-bound receptors, the G-protein coupled receptors. Second, it is an example for a membrane transporter. This is a class of proteins which is essential for all organisms. Bacteriorhodopsin generates a 10,000-fold difference in proton concentration on either side of the membrane. Third, it's handy, meaning that it has several properties that make it easy to work with this protein. The transport process can be easily triggered by light, which enables many kinds of experiments. It is easily obtained in large quantities and unusually stable. The structure of Bacteriorhodopsin is shown here. It consists of several transmembrane helices, and nearly all of the protein is embedded in the membrane. The membrane surface runs approximately horizontally for the top leaflet here and for the bottom leaflet here. The protein contains also a chromophore, shown in green. The word chromophore is Greek and means color bringer. The chromophore makes Bacteriorhodopsin absorb visible light so that it appears purple. The chromophore in Bacteriorhodopsin is called retinal. The retinal forms a covalent bond with a lysine residue in helix G. This connection is called Schiff base. The chromophore retinal is also present in the protein rhodopsin in the retina of our eyes. This protein is our primary light receptor. After absorption of a photon, retinal in Bacteriorhodopsin changes its conformation from all trans to 13 cis. This is a rotation around a double bond, which is facilitated in the excited state because of a changed electron density distribution in the molecule. This isomerization is an example for a photoreaction. The photoisomerization reaction in Bacteriorhodopsin proceeds with high yield. This is in contrast to model studies in methanol, where retinal linked to a protonated Schiff base forms cis isomers after light excitation only with a probability of 15%. The by far dominating conformer in methanol is the 11 cis conformer. Thus, the protein environment increases the yield of the photochemical reaction and determines the conformation of the photoproduct. The isomerization of the retinal is transmitted to the protein environment and starts a cascade of reaction steps which transport one proton across the membrane. This involves changes in protein structure as well as protonation and deprotonation of several groups. Steps are indicated here and will be discussed later. The photoreaction of Bacteriorhodopsin proceeds via several intermediate states. The intermediates are designated by capitals. Br is the ground state that absorbs a photon. It is also called resting state. In Br star, the retinal is in an electronically excited state. The photocycle proceeds then via intermediates J, K, L, M, N, and O back to the ground state. In O, Br and Br star, the retinal is in the all trans conformation. In all other states, it is in the 13 cis conformation. The Schiff base is deprotonated only in M, as shown here. The intermediates have different colors. In other words, they have different absorption spectra in the visible spectral range. The indices indicate the absorbance maximum in nanometer, like here for the J intermediate. Therefore, the intermediates can be followed by time-resolved spectroscopy, and that has been done intensively. Why does the color of the retinal change? The color of retinal is tuned by electrostatic interactions with the protein environment. These interactions change during the photocycle, and therefore retinal's color changes also. So one aspect of the color change is that the protein environment influences the color of the chromophore. In Bacteriorhodopsin studies, researchers take advantage of this effect to study the protein. In our retina, nature uses the same effect to enable color vision. 3 different types of rhodopsin proteins with the same chromophore retinal have different absorbance maxima. They are in the blue, red and green spectral region because the different proteins provide different environments for retina. In the following slide, I will show you the spectra of the Br ground state and of the M intermediate. So please remember that Br in the ground state is purple, while the M intermediate state is pale yellow. Br absorbs in the visible spectral range, which is entirely due to the chromophore retinal. The rest of the protein absorbs only in the ultraviolet, not in the visible spectral range. The absorbance maximum in the ground state is near 570 nanometer. Yellow and green light is absorbed, while blue and some red light can pass. Therefore, the color of the Br ground state is purple. In solution, a model retinal compound with protonated Schiff base absorbs at 440 nanometers, at much shorter wavelengths than retinal in the Br protein environment. The additional redshift to 570 nanometers for the all-trans chromophore originates from electrostatic interactions and from the planarity of the retinal backbone. This is another example for the influence of the protein environment on the color of the chromophore. It defines the electrostatic interactions and restricts the conformation of the chromophore. The largest spectral change in the photocycle occurs when the M intermediate is formed. The M intermediate absorbs at 150 nanometers lower wavelength than the ground state and has an absorption maximum near 400 nanometers. Therefore, it has little absorption in the visible spectral range and is nearly white. But not quite, because some of the blue light is absorbed. This makes the M intermediate appear pale yellow. These spectral differences can be exploited to study the photocycle of Bacteriorhodopsin. For example, one can find out how fast the M intermediate forms. For this, one measures the absorbance at 400 nanometers, for example. Such a measurement will show an increase in absorbance when the M intermediate forms. By repeating such measurements at different wavelengths, optical spectroscopy has detected the intermediates in the photocycle and characterized the number and kinetic properties of them. One can then continue and study the effect of mutants on the photocycle. In this way, residues can be identified that are important for particular reaction steps. In addition, optical spectroscopy has the important role of assigning properties measured by other methods to particular reaction steps in the photocycle. For example, time-resolved infrared spectra reveal changes of retinal conformation and protonation and deprotonation of amino acid side chains. When we correlate this information with spectra in the visible spectral range, we can find out at which step of the photocycle these changes occur. Similarly, when proton uptake from and proton release to the bulk are measured, these events can be correlated to the photocycle using the spectral properties of the Br intermediates. Optical spectroscopy has also been used to assess the functional activity of Bacteriorhodopsin in crystals, which is an important prerequisite for time-resolved X-ray crystallography. Of particular advantage in these studies is the relative simplicity of the visible spectroscopy experiments, the high time resolution, and the high sensitivity, which means that only little protein is consumed. Finally, after many years of research, the following view of the photocycle has emerged. The first steps are absorption of a photon, isomerization of the retinal from all trans to 13 cis, and return to the electronic ground state. All this happens in 0.5 picoseconds in the transition from Br to the J intermediate. The retinal is in a strained conformation that relaxes somewhat in the steps to the L intermediate. Next, the proton moves from the shift base to aspartate 85. Remember that the shift base is the linkage between the retinal and lysine 216 of Bacteriorhodopsin. The proton transfer takes place because the pKa of the shift base decreases by 7 units upon retinal isomerization, and the pKa of aspartate 85 increases to more than 10.5. A proton from a protonated cluster near glutamate 194 and glutamate 204 acidifies the extracellular medium. All this happens in the L to M transition. Then, in the M to N transition, a proton moves from aspartate 96 to the shift base. This restores the original protonation state of the shift base. The proton transfer takes place for two reasons. First, the pKa of aspartate 96 has dropped from larger than 11 in the ground state to about 7.5 in the N intermediate, and is now lower than that of the shift base, which is about 8. Second, a chain of water molecules start to form in the M state that serves as intermediate proton acceptors. In the N to O transition, aspartate 96 becomes reprotonated by a proton from, amongst others, aspartate 38. The retinal isomerizes back to the all-trans state. Finally, in the transition from O to the Br ground state, the proton of aspartate 85 moves via a proton channel to the extracellular release site near glutamate 194 and glutamate 204. As you can see, it is not the same proton that is transported all the way from the intracellular side to the extracellular medium in one photocycle. Instead, several protons move towards the extracellular side, each covering a fraction of the total distance in different regions of the protein. The mechanism of this protein is just amazing. There is one energy input into the system, which is the absorption of a photon. This makes the retinal isomerize, which is aided and guided by the protein environment. Isomerization of the retinal leaves both the retinal and the protein environment in a strained conformation. Both try to relax to the lowest energy conformation, which is the ground state. Often, such relaxation processes just lead to heat transfer to the environment. Not so in Bacteriorhodopsin. Its relaxation process proceeds via a number of tightly controlled steps that couple energy relaxation to the pumping of a proton. As an illustration, imagine the following. An initial energy input makes you stand up. Then you relax by sinking down into a comfortable sofa and finally adopt the resting position. At the same time you do useful work. Hard to imagine, but this is exactly what Bacteriorhodopsin does.